Okay, so we're really just going to talk quickly about membranes, right? We've already kind of dealt with these concepts, because that's what our lab dealt with. So pretty straightforward. So why are membranes selectively permeable? What are the role of phospholipids, proteins, carbohydrates, and membranes? How water will move if a cell is placed in isotonic, hypertonic, and hypotonic solution. And the key thing, electrochemical gradients. Right, so these are kind of the key concepts associated with this chapter. This is definitely a bigger concept chapter than just a cell organelles. So when you have your test next week, it's going to be heavy on this and light on organelles. We'll just have some basic stuff then. All right, so here's our old friend, the phospholipid. Right, we have one tail is saturated, one tail is non-saturated or unsaturated. Right, so we have our double bond right here, causing it to be bent. What did we said when we had unsaturated fatty acid tails? What did that material tend to be at room temperature? Liquid, which is weird, right? Why would our membranes be liquid at room temperature? But technically they kind of are, at least for right now. We're going to add some other things in here. Right, so we're familiar with our phospholipid, the hydrophilic head, the hydrophobic tails, and so forth. These are the key components that you should just be familiar with when it comes to the components of our membrane. Right, it's this mosaic of a lot of different structures. Right, we have our phospholipids. We have integral proteins, proteins that pass through the membrane. We have peripheral proteins, proteins that are on the surface of the membrane, either on the internal or external surfaces. We have carbohydrates that play a role as well. We've mentioned carbohydrates like the oligosaccharides. Right? They play a role in determining cell identification. So if I have A blood or B blood, right, the carbohydrate chain would either be an A carbohydrate chain if you have A blood, a B carbohydrate chain if you're B blood, or both if you're AB blood. Yes?
Yeah, just a quick question on blood. Like, isn't there also like a RH?
There's a whole other. There are a number of different factors that you could do to analyze blood type. The simplest one is simply the AB. Then the RH factor is a positive or negative. It's a rhesus factor. So that's even when you go further down from there, there are other characteristics you could look at as well. But for blood transfusions, really just the type and then positive, negative are the only ones that really matter truly for blood transfusions. However, if you want an organ transplant, you need to be a match with more indicators. Okay, so a couple of the carbohydrates are glycolipids or glycoproteins. Glyco meaning carbohydrate. Carbohydrate attached to lipids, carbohydrates attached to proteins, and so forth. So taking a look at our structure here, right? It's this complex structure, this mosaic of all these different materials. Do you need me to go back for a second?
I'll just pull it up on here.
Okay. So we have all of our phospholipids. We have our glycoproteins, right? Identification cards, the oligosaccharide chain. We have our glycolipids, right? Same thing, just attached to a lipid molecule. We have our integral proteins, our peripheral proteins. Maybe possibly this is a peripheral here as well. Definitely peripheral here. So there are a number of different components. Now, your membrane is trying to be fluid at room temperature, which is kind of bizarre, right? It seems like it has to be more of a solid structure. But we do solidify it. Proteins are solids. So by adding in all these proteins, it's helping to solidify the overall membrane as well. And then the other key structure there is cholesterol. Cholesterol is vital. We need to have cholesterol. Because we need to embed cholesterol into our cell membranes, which helps to solidify the membranes. So cholesterol, although too much in your bloodstream is a bad thing, because it can cause clogging plaques, you do need a certain amount of it to be embedded in your membrane to help solidify your membrane. Couple interesting things, right? If you look at an organism that lives in colder environments compared to an organism that lives in warmer environments, you can analyze their membranes for cholesterol levels. Organisms that live in warmer environments are going to have more or less cholesterol in their membranes. They're going to have more, right? Warmer environment means trying to be more viscous, so it tends to have higher levels of cholesterol in their membranes. Organisms that live in the Arctic are going to tend to have lower levels of cholesterol. That kind of makes sense. Yes? Absolutely. So other couple key things here, right? We have our attachment to our cytoskeleton. which attach to a lot of these integral and peripheral proteins, and attachment to the ECM, the extracellular matrix. A couple of interesting things when we look at cell membrane. Lateral movement, so a phospholipid kind of switching places with the phospholipid. Actually, again, it's very fluid. It happens about 10 to the 7 times per second. So your phospholipids are moving all over the place. They're very, very active. However, vertical movement is a lot less likely to occur. Why is vertical movement a lot less likely to occur? Exactly. The hydrophobic tails are exposed when you have the vertical movement. So therefore, there are forces in play not really stopping that movement from occurring. All right. So just kind of key things. Now let's just kind of focus on the actual proteins themselves. This is pretty straightforward, right? When we look at proteins, there are certain functions of proteins, right? So we're talking about integral or peripheral proteins. Right? We definitely know that they can be used for transport. Transport, enzymatic activity, signal transduction. Don't worry about that right now, but it's pretty easy. It's kind of like playing a game of telephone. You'll know a lot about signal transduction as we go down the road. Cell-cell recognition, intercellular joining, attachment to cytoskeleton, and ECM. So if you want to keep on this slide and take some notes, this slide is just showing the same thing in images. So transport, we have transport proteins, right? Passive transport, diffusion, facilitated diffusion. We also have active transport being shown here. A lot of times some transport can go through the proteins. Enzymes, our proteins are enzymes. So some of these integral proteins or peripheral proteins might be enzymes just being held on the surface, carrying out either reactions internally or reactions externally, either way. Signal transduction is just kind of relaying a signal from outside the cell to inside the cell. It's simple for now. We're not really going to go in detail yet, but we'll talk about that. Here's our glycoprotein for cell-cell recognition. Right? A skin cell wants to know if the cell next to it is another skin cell or some other type of cell. So cell-cell recognition, if it's a foreign invader, these things would be considered antigens. Right? The antigens are the markers in cells, so this would be an antigen right here. Cellular connections, right? What would this be an example of? 3 types of cellular connections. Could be a gap. It's not really showing it, but it could be a gap junction if there was a little channel here. Could be a adhering junction or desmosome, or it could be a tight junction, any of the above, but they're all types of cellular connections. And then the last one is simply attachment to the ECM. So these are all key functions of our proteins, just something to keep in mind, always kind of talking about the functions and so forth. Just kind of mentioning the glycolipids and glycoproteins, right? These are the identification cards. These are the antigens outside your cell membranes, on the outside of your cell membranes, allowing cells to recognize each other. We just kind of highlighted they can vary among species, individuals, or even individual cells. Right? So one skin cell might be identified and another skin cell as being slightly different. So there's a lot of variation with these glycoproteins, with these antigens. This right here is nothing new. It's just kind of a nice picture to put it all together. But your cell membrane is complex. Well, how do we make your cell membrane? You simply make it through the endomembrane system. That's it. So when you kind of follow that process of protein synthesis that occurs on the rough ER, you just continue to follow this process. Here's our rough ER. Here's where a ribosome was. Now remember the video of the protein being made and traveling into the rough ER? Well, picture that protein just not completely passing through. Picture that protein remaining partially inside the rough ER and partially outside the rough ER. What do we have at this point? We now have an integral protein. All right, so this little dumbbell is representing your transmembrane protein or your integral protein. So it gets formed right here at the rough ER and simply gets modified and moved around as it gets to the membrane itself. So just kind of showing, like, how do we get this complex cell membrane? Well, it's just made through what we talked about before, through protein synthesis and the endomembrane system. Pretty straightforward. let's say I need to add on to a specific oligosaccharide, right, to make a certain glycoprotein or glycolipid. Guess what? That can happen in the Golgi apparatus, right? So just kind of relating that process to the endomembrane system. Okay. We didn't talk about all this yet, did we? No. Okay. My freshman class, because you guys are already catching up to them, I start with macromolecules with them, so you're catching up. So I do a similar lecture with them, pretty much the same thing. And their heads spin. But I've done this lecture so many times, I feel like I've already given it to you. Okay. So, real quick, just kind of relaying some things here, looking at structure. With this, wouldn't have been good to draw this thing. We're going to label just some different regions of our integral protein. Okay. Now, these proteins do not look like they're well attached to the membrane. They look like they can pop out pretty easily, right? Like what's actually holding them in place? Well, when we start thinking about the actual amino acids and their R groups, it helps us to understand how they're being held in place. So here's this region of the protein up top, region A at the top or bottom. There are going to be specific types of amino acids there. What type of amino acids are we going to have in region A? Polar, right? Nice and easy, right? So you're going to see more polar amino acids there, where region B, we're going to see non-polar interactions, right? And those interactions are what hold that protein in place. If it starts to kind of squeeze upwards, those non-polar R groups on the amino acids are going to start coming in contact with water, and it's going to be pushing itself back down. And then the last one is, if this was a protein channel, bless you, we would figure that small non-polar molecules could move directly through the cell membrane themselves. So what type of molecules would actually go through a protein channel? Very good. Larger, polar, or charged. Right, such as ions, sodium ions, potassium ions, chlorine ions, and so forth. Whoa, something's going on. All right, so pretty straightforward, just kind of looking at polarity. We keep coming back to it. It's discovery. Be careful. This is just it's reactive, so be ready. It gets hot fast.
It's red dye. I knew it. You're good at that, but your face is like serious.
All right. So really, what we care about in this chapter is we care about movement across the membrane. We care about diffusion. All right, diffusion, a process that occurs naturally in our world. What realm of physics/chemistry deals with the movement of molecules? Fluid mechanics, and what laws dictate it? Starts with a TH. Thermodynamics. Very good. Okay. What's one of the first laws of thermodynamics? We can't create or destroy matter or energy. Very good. What's the next one? Starts with an E.
Energy.
Not energy. And entropy. Entropy. All right, what is entropy? What is it? The amount of randomness. The amount of randomness, right? Randomness in terms of molecules. if you were to use entropy to talk about our universe, what's happening with entropy in our universe all the time? Constantly what? Increasing, right? So we're always moving towards more entropy, right? There's more and more chaos in this world. Right? Entropy is a measurement of organization, and we're always progressing towards more entropy, as a universe, as an entire system. Okay, entropy is really what's going on when we look at diffusion, right? When something diffuses, it's going from an area of high concentration, high organization, to areas of lower concentration, less organization. Right? So the highly organized drop, when it hits the water, would be something that has very, very low entropy, and it's going to progress and increase entropy, more chaos, more disorder by spacing and getting as far apart as possible. Okay. One quick thing on entropy. Do we possess high or low entropy? Humans, organisms. Low entropy, right? We possess low entropy. Why do we possess low entropy? Because we are highly organized. We're highly organized, right? All of our molecules are put together. We're highly organized, right? A flower is highly organized, right? Their molecules are all in the right place, everything. If the universe is always progressing towards increasing entropy, how do we prevent entropy from occurring? We organize ourselves, right? Your atoms, they think about, okay, let's organize ourselves. How do you organize yourself? What do you use to organize yourself? What do you need? Food. We need energy, right? In order to maintain, right, very, very low entropy, you need to eat. You need energy, right? If you stop eating, you are not going to maintain low entropy. You're going to start becoming disorganized. You're going to die. Right? And your molecules are going to be broken apart. You're going to decompose, right? So we maintain very low entropy, a state of organization, by eating all the time. Just kind of a side note. Okay, so diffusion follows the laws of entropy, right? Diffusion is simply molecules going from an area of high concentration to an area of low concentration. Very simple, right? I'm going to light a candle over here. Just got my candle from Yankee Candle Company. What scent do we want it to be?
Vanilla.
Apple. Vanilla. Okay, vanilla. It was lavender the last two classes of my bio. They both chose lavender. All right, so it's a vanilla candle, right? Light the candle on fire, right? And as it's burning, the oils are being released. They're becoming gas, and they're highly concentrated right here. It's going to become very vanilla right here. It's going to smell good. If there was no air circulation, if we did not move at all, would you eventually smell vanilla on the other side of the room? If there's no air circulation, we don't move, we don't waft it around, would you eventually smell it on the other side of the room? It would take time, but you would. Why? Because these molecules are banging into each other, and they're in a very, very organized state. They don't like that. They want to become disorganized. So they bang into each other, and they start spreading out, right? Higher and higher entropy, more disorganization as they spread out around the room, and eventually you'll smell it over there. So it's just a natural law of nature, right? Everything goes from ordered states to disordered states. Taking it to diffusion, molecules always go from a high concentration to a low concentration, right? I didn't stir this or anything. You can see it kind of diffusing here over time. And again, I didn't stir it, so it's just doing it naturally. Okay, so what we really care about, not just diffusion in a beaker, we really care about diffusion across a membrane. Couple quick things. So, diffusion simply is going from an area of high concentration to low concentration. That is known as going down a concentration gradient. The concentration gradient is like a hill. High concentration being the top of the hill, low concentration being the bottom of the hill. Right? If you let the ball go, it'll naturally, with no energy input, roll down the hill. Okay? To a lower energy state. And what happens is you finally hit a point of equilibrium, but it is not a static equilibrium, it's a dynamic equilibrium. Right? If I have a cell that's 2 molar sucrose in a solution that's 3 molar sucrose, eventually the concentrations will level out. At that point, does water stop moving across the membrane? No, it keeps moving. You're in a state of dynamic equilibrium. It's kind of a net movement of zero. Some water goes one way, some water goes the other way, right? We're in dynamic equilibrium. Okay, so we're talking right now, and just a kind of a good thing to write down in your notes. Where did I put my marker? There. We are talking right now about movement that does not require energy, so we are talking about passive transport. The first example of passive transport is simple diffusion. Simple diffusion is just as a sound. It's diffusion simply across the membrane itself. So, going from areas of high concentration to areas of low concentration. Something is wrong with this slide, though. What's wrong? There's something up here that's wrong. See how smart you are. Yeah, there are no proteins in the picture, but that's okay. You're right. That's not necessarily what we're looking at. There's something wrong. What is it stating water is? Is water lipid soluble? No, oil doesn't mix with water. So water's not a lipid soluble. Water actually has problems getting through the membrane itself. It has to really move through aquaporins in real cells, although the water can move through the membranes we're dealing with, which you'll see tomorrow. But water's not a lipid soluble, right? Water's water. It's very polar. However, O2 and CO2 are non-polar lipid soluble molecules. So if I have a high concentration of oxygen in the extracellular fluid, oxygen is going to be moving into the cell. It's going to be diffusing into the cell. If I have a high concentration of CO2 in the cytoplasm, CO2 is going to be diffusing mainly out of the cell. You always get movement in both directions, but the net movement will be down the concentration gradient. Easy enough? This is simple diffusion. Next one we have is facilitated diffusion. So passive, our examples are bless you. I didn't sleep last night. I don't know why. What we have is facilitated diffusion, which is involving a protein channel. Here we have a channel protein. Notice it is open. The molecules are simply just going from high concentration to low concentration. Does it require energy? No, it's passive transport. Here we have carrier proteins or gated channels. So carrier protein, sometimes known as a gated channel. With a gated channel, could there be some energy needed to open or close it?
Yes.
Possibly. It doesn't have to be, but there could be. Does there need to be any energy used to move the molecules from a high concentration to a low concentration once it's open?
No.
No. So it still is passive transport. Both of these are examples of passive transport. The last kind of example of passive transport is which is what we dealt with our potatoes, right? We had the movement of water. Sucrose was too large to move across the membrane, so we moved water instead. With osmosis, right, we have some certain terms here. Osmosis is the diffusion of water. Tonicity is the ability of a solution to cause a cell to gain or lose water. Don't really worry too much about tonicity. We'll talk more about water potential. Is a better word for that. If there is a water potential, that means water is moving. Water has the ability to do work. Isotonic, hypertonic, hypotonic are three kind of terms, right? Isotonic would be equal concentrations. Hypertonic solution would be a higher concentration in the solution. Hypotonic would be a lower concentration in the solution compared to the cell. So here are the definitions. Right, here are kind of the images we can talk about. So here we have our isotonic situation. We have equal concentration in and out of the cell. Notice water's still moving, it's just moving in a net zero situation. Notice a red blood cell, animal cells, prefer to be in an isotonic solution, an isotonic situation. Why? They don't have a cell wall, right? So they want to have this balance. They want to be in equilibrium. Plants do not. Plants want to have water constantly being pushed inward. Why? Because it gives rigidity to what structure? What structure? The cell wall, right? Helps it to stand up. So a plant that is in an isotonic situation, that plant is something that's wilting. It's not standing up strong. All right. If I put an animal cell in a hypotonic solution, right? Meaning the high concentration is inside the cell or in the solution. Where's the high concentration? Remember, we're talking about the movement of water, right? We're talking about like sucrose that can't really move across the membrane. The high would be here. It's hypotonic, always referring to the solution. So the low would be there. How do we try to lower the concentration inside the cell? If I can't move sucrose out, what do I move in? Water moves in, right? So we get a net movement of water in. Therefore, it's going to cause that cell to swell. Animal cells don't do well with that, right? They're going to burst. Their membranes are soft. They're weak. Plant cells, however, like that because they build up what's known as Turgor pressure, right? The outward pressure on the cell wall, giving it the strength to stand up tall. Okay. So this is in a hypotonic. Again, hypotonic solution. We're always referring to the solution unless the question somehow reverses it to talk about the cell. It's always referring to the solution. Now we have our hypertonic. So that means the high concentration is here, the low concentration is here, which causes our cell to lose water, causes it to shrivel. This one would actually kill both. It could kill this cell, but it'll definitely kill the plant. Because what happens is the cell membrane, it goes through something plasmolysis. Which is the cell, the central vacuole is losing water, losing fluid. So the cell membrane is starting to shrivel. Well, guess what? The cell wall doesn't shrivel. The cell wall is rigid. So the cell membrane starts to pull away from the cell wall, which actually kills the plant cell. And just kind of like finishing up on this. So turgid is what they want. Flaccid is what you have when it's bad, when in terms of it's wilted. And then plasmolysis is when you have the cell membrane actually pulling away. So here's kind of plasmolysis in a plant cell. This was the last piece of your lab, but we didn't do it. So they're going to add what's going to be a salt solution. So it's tough to see, but you will see it. You can see the space here. Oh, it's starting to pull away. It's amazing, isn't it? But you can definitely see the cell membrane pulling away from the cell wall, which actually causes the plant cells to die. And if they die, they die.
Why does the cell membrane pulling away from the